A few quick housekeeping things. Firstly, can I just check that everyone can in fact see a screen that says understanding the etiology of reproductive aging and its links to broader health? I'm seeing some nodding. Secondly, I'm going to cover quite a lot of varied topics in this talk. This is going to be very much an overview of a range of different things I do. So I'm aware that questions may appear to you as you're going along. Feel free to ask them in the chat in Zoom. That's probably the best way to get them, and I'll try and answer them as I go along. I will, of course, leave some time at the end to answer any additional questions or any general points that you've got at the end. Okay, so that was the sort of housekeeping over. So I'm going to be talking to you about the reproductive aging and its links to broader health. So I'm going to be talking a little bit about puberty timing, a bit about some other reproductive timing, and how we might use these, some of the methods that we use to understand them, and both their biology and their links to health. So first, some overview of the sorts of things I look at. So I look at puberty, I look at age of first birth, I do some work with sex hormones, PCOS, some other fertility measures, and menopause. So those are my sort of core phenotypes, and then I use those to understand their links to type 2 diabetes, particularly. We're in a unit that focuses on type 2 diabetes, but also to obesity, other metabolic diseases, cancers, and a little bit about some of the sort of more behavioral phenotypes that some of these might impact as well. So a little bit of a history lesson to start off with. So this is about the first study of puberty timing in a sort of GWAS type measure. It was done on 18,000 women. And it asked them to recall their age of menarche. So when we're talking about puberty, we're almost always assessing adult participants, and we're asking them to recall an event that may have happened 50, 60, 70 years ago. So there was some worry in the field at that point about how good or reliable this was going to be, but actually it turned out to be, certainly for a GWAS type study, sufficiently reliable. And they were able to find 2 hits in the genome-wide study, one at Lin28B and one at ZNF42. And these have an effect of about 5 weeks of changing the age at which you go through menarche. It was a reasonably popular thing. 3 other papers at the same time all reported Lin28B. Some of them had found the other signals, some of them hadn't. But it was certainly an area that stimulated other people to look at this as a potential avenue for genetic research. And now, in 2017, we published, this is the most recent study, we published a study of over 300,000 women, rather than just 2 loci. We found 400 robustly associated loci. And now, rather than just looking at about 5 weeks, we're looking at loci that have an effect that varies between about 1 week and 1 year in some cases. That's for one of the rarer variants. And in total, these explain about 25% of heritability. So we've got a good grounding to then explore other downstream analyses using these genetics. We've got a good understanding and many SNPs that explain puberty timing. So if you've got all of those 400 loci, what can we actually do once we've got a SNP? And this is possibly the nicest case example that you'll ever have. So this is a study. We've got 3 hits here. I don't know if you can see my mouse, but we've got 3 hits here. Sort of the orange in the LD, the green in the LD, and this pink in the LD. So 3 independent hits of this region. And they're all around this gene called AMH. Now this is a GWAS for AMH levels. So this is really nice. We found AMH. We're pretty sure that we understand what the biology is going on here. We've got a signal of this gene. This explains the expression of this particular protein. It's a sex hormone that regulates, well, it actually regulates turning into a boy or a girl, but it's also important in puberty timing as well. So that's a simple way of neatly linking a SNP to immediate biology that's got going on there. However, often you'll find this isn't the case. You actually have many more SNPs in this region, or sometimes there's no genes at all. So it's hard sometimes to link a SNP to a particular piece of biology, but there are other things that we can do. So we can look to see what have happened in other studies that have used different methods to find this region. So this is a particular region which we're TACR3. So this is a puberty timing variant that we found. The common variant that we found was at the top row of that table. Occurs at an allele frequency of about 15%, and it has an effect on puberty of about 2.5 weeks. Probably not something that anybody would notice. If, however, you've got a much rarer variant, which is a loss of function, and if you've got 1 copy of those, something we can pick up in exome array type studies, then there's an effect of about over a year. So that's not necessarily something that you might necessarily go to a clinician about, but if you're a child at that age not going through those stages of development compared to your peers, that's something that might potentially have a sort of sociological effect on you. So it's important to remember that reproductive diseases and conditions are not just about the specific clinical disease, but they also have an important societal pressure as well. And then if you're homozygous for that loss of function, it's about 1 in a million people, then you have delayed or absent puberty. So while you probably wouldn't consider like a change in puberty timing of a couple of weeks to be something that was clinical, if you're 30 and you haven't gone through puberty, then that's something which is actually a clinical condition, and understanding the biology that underlies that is important. So we can study data from different types of studies, but all of the same underlying condition, or we can look at similar conditions. So this is a set of variants that have been identified for voice breaking. We're comparing them to the age of menarche. And broadly, you can see that actually the majority of SNPs that are discovered for one or other are general puberty-specific variants. So the things that underlie puberty in a biological way between boys and girls is basically the same for the majority of genetic variants that we're looking at. There's a few, so this retinoic acid receptor up here, and this leptin receptor here, which look like they have particular effects either in girls or in boys. So it's something that's specific to males or females. And we still don't properly understand what it is that's going on here. There's this variant SIM1 down here in the corner, which appears to have divergent effects in boys and girls. So in boys, it makes you have your timing of puberty measured by voice breaking much later, whereas in girls, it's something that's happening much earlier. So it's this sort of odd bidirectional effect in boys and girls. And honestly, we don't really understand what it is that's going on at that locus, but we can hopefully try and add other information from other phenotypes on to help us tie together this story. And we can do this both for phenotypes that are obviously closely related, like puberty timing in boys and girls, or we can look across a much wider range of phenotypes. So this is data from the Oxford Big Study, and I put in one of my favorite variants in a gene called CADM2. Now, this was discovered for a study that we did of a number of children. It's down there, just passes the significance 9, wasn't the strongest association in this region, but it was very strong in the study that we were doing for a number of children. But also you can see up here that there's a large number of body composition type variables. It seems to be something that increases your body size and affects the number of children. And then potentially this was more salacious, but actually you've got things here like risk taking. And then if you look at the other variables that are associated with that, alcohol intake, ever smoked, and ever drive too fast on the motorway, these are almost certainly downstream variants of the risk-taking variant, which is very strong. So we may here have a situation where we've got something that we think is behavioral, risk-taking, and that may then have an effect on other variables, such as driving too fast on the motorway. And driving too fast on the motorway is a major source of disability and death in people. It's a cause of major accidents. And also ever smoked and smoked alcohol intake. These are two things which are probably health degrading as well, but also potentially has an effect on number of children. There's potentially a behavioral change in the number of children that you have. So by looking across a number of different phenotypes, we can potentially try and tie together a little bit of an idea about what these SNPs are doing more broadly. The other thing we can do is we can see whether or not these associations hold up in different populations. And I think one of the things that we've been quite pleased to have done is to look at a study of almost 70,000 women in the Japanese biobank. We considered both menarche and menopause here. And you can see actually looking at the heritability estimates between these two populations, there's actually some differences. So the heritability is considerably higher for both menarche and menopause in Europeans as they are in Japanese populations. In your populations, they're also a younger age. So the age distribution of the timing of menarche in European populations versus East Asian populations, we'll come on to that a little bit later in the talk, is actually markedly different. So there's definitely an environmental background that these SNPs are operating on that's different. And if we consider like the specific SNP, so this is a SNP that we found in MCM8, which is quite strong in Europeans. It's one of the first signals that was discovered for puberty timing. In Japanese populations, it's not there at all. Now, for this particular case, it's not there because it's a monomorphic population. So this SNP simply doesn't exist in each East Asian populations. But as you look in the table below, there's 4 other SNPs here for menarche and for menopause, which have a markedly different effect in the European populations versus the Japanese populations. And some of these are about the significance of these is due to the frequency of these variants. So for example, if you move from a frequency of 40% to a frequency of less than 1%, you're going to get far less significance. You just don't have the sort of power. But equally, there are significant variants here, such as a move from 51% to 25%, which is probably not simply a power situation. It's potentially that there is a gene-environment interaction here, which is particular to the environmental background of puberty between these two populations. So we can't pin it down particularly to a specific environment, but we could say broadly that there's likely to be a change between these populations affecting what's happening at this SNP particularly. Okay, so that was a little bit about SNPs. But a lot of that was about what a SNP is doing, particularly in terms of its association with other phenotypes. Can we say a bit more broadly about stuff and its relationship to biology? And the first thing is just, again, a way of leveraging genomic information to look across phenotypes. So rather than just looking at a single SNP and seeing its specific associations across different phenotypes, look to see what a genome-wide leverage tells us this. So effectively, this is a method that compares two phenotypes, looks at the genome-wide information and sees whether the beta estimates between those two are correlated. And it takes into account the LD between SNPs and a genome so that we're not double counting here. But it effectively boils down the shared biology of these SNPs as represented by the genome into a single value. And effectively, you end up on a 1-to-0 scale, a genetic correlation, if you will. And if you look across these graphs, you can see a number of sets of phenotypes that come together. So there's the one in this top corner here, which includes BMI, childhood obesity, fasting glucose, type 2 diabetes, and coronary artery disease, which is clearly a sort of a metabolic type set of variants. So there's a core of shared biology between these outcomes that is evidence in their underlying genetic architecture. Equally, we've got a set of diseases, Crohn's disease and ulcerative colitis, so diseases, autoimmune diseases of the digestion, where there's a common core going on there. We've also got a set of birth weight and birth length type variables, particularly influencing your size as a child. There's a commonality between there. And then right down there at the bottom, we've got these depression, bipolar, and schizophrenia. Again, strong correlations between these three, suggesting that despite these quite different presentations of mental illness, there's a commonality in some of the underlying genetic architecture of these traits. So that's a little bit about the links between different diseases and how they might have some sort of shared biology. But we can also use this method of trying to look at correlations between two genomic databases to understand a little bit about which particular tissues are being acted on here. This is potentially a little bit out of date. You might have done something similar in some of your other courses with a slightly more up-to-date method, but broadly, a lot of these are about trying to partition the heritability into particular types of tissue. So here, it took the GTEx data and it took the 53 tissues from those and ranked the genes that were were most distinct across those different tissues. So you ended up with a set of genes which were particular to those particular types of tissues and not seen anywhere else. And then we use this LD score regression rather than just looking at the betas for a particular phenotype, but we looked at those betas for an expression of those particular tissue-specific SNPs. And that meant that we could effectively identify a genome-wide measure of how likely these particular tissues were to be underlying our disorder. And as you can see here, so this is the date from puberty timing. The puberty timing is almost entirely in the brain. So all of the tissues in pink there, the ones that are strong and significant, are those which are at the brain. And I'll come on to this a little bit later when I'm talking about one specific example. But overall, the hormonal control of puberty is something that's absolutely initiated in the brain and then feeds down to the rest of the body. It's something that starts there. Sometimes you get kind of lucky. So that was an example from puberty about how we were particularly focusing on sets of tissues. We weren't necessarily saying anything about pathways there, but we were at least specifying that it was the brain that was important. So this is an example from menopause, and we got kind of lucky for menopause. We started off and we were looking down the gene list of what we identified here, and without having to actually needing to do any pathway analysis, we were seeing common DNA repair variants that were coming up very strongly. So BRCA2, BRCA1 is now coming up, MCM8, CHEK2. And we began to see quite clearly that about 2/3 of the variants that were identified for menopause were strongly related to double-stranded breaks. And this was visible both in humans, in cellular work, and in mouse work. So we began to build this picture that actually menopause is primarily driven by the body's ability to maintain oocytes. So you can see here, this is effectively a curve of the number of oocytes over a woman's lifetime. But actually, the number of oocytes a woman has peaks prior to birth, and then there's a decline after that. There's effectively a half-life of oocytes. And many people had previously said that perhaps oocytes were lost through ovulation. Perhaps that was the principal driver of when you had menopause, because menopause effectively happens when you get to a critical mass of oocytes that's very small. But actually what we show here is that the peak is potentially important. There may be some about the number of oocytes you have at the beginning, but actually the major factor is the rate of decay of those oocytes. and that's the single thing that determines DNA damage, to the extent that actually the genetic score for DNA damage is the strongest single predictor. It's stronger than smoking at this moment to predict age at menopause. So this is the overwhelmingly important factor. Unfortunately, not many diseases have this, of the complex diseases that we've been talking about here, have this overwhelming, obvious single factor. So this is slightly unique in terms of the information that we have for menopause. To compare that to puberty, and this is the ones where we certainly know a lot less about what it is that's going on in terms of the biology here. But one of the variants that we do know a little bit about is this variant called MKRN3. So MKRN3 is effectively a break on the GnRH sequence. So this cartoon diagram here is effectively what's going on in terms of the hormonal control of puberty. So GnRH is secreted, and that simulates LH and FSH to then go to, in this case, the ovaries, but equally to the gonads of the male. But by having MKRN3, it acts as a brake on this GnRH secretion. So high levels of MKRN3 exist in early life, and then they drop off. And at that point, GnRH increases, and then that starts the cascade of hormones that result in puberty. And we can see that in humans here, so this red and green line here, and also in mice. Mice obviously have puberty considerably earlier than humans, so it's something that only really exists in the early ages of the mouse. And loss of function variants in humans have precocious puberty, so they go through puberty very, very early. It's not necessarily a disease that we see particularly commonly, but again, when I was talking before about how puberty might be a clinical disease, if you're going through puberty, in some cases, with some conditions, 2 years, 18 months, that's something that is clinical, and you're going to need to go to a doctor, or at least be taken to a doctor in order to understand. So that was a little bit about how we might use this information in terms of understanding the biology, or some of the underlying biology of some of these conditions. Now I'm going to talk to you a little bit about how can we use this information to inform epidemiology and public health. And I work in an epidemiology unit, so this is much more of my sort of core day-to-day. And I think this is definitely an advance in human genetics that is particularly important in terms of understanding how we can help to inform the study of diseases. So I'm hoping that you will at some point have come across this idea. So Mendelian randomization is how we can use SNPs associated with our particular outcome of interest as a proxy for a disease. And we hope that, similar to a randomized controlled trial, that our exposure or intervention of SNPs here is randomly allocated with regard to many of our potential confounders. That's not always true, and I'm going to come on to a couple of examples which may demonstrate that. But this is the underlying logic that we have here. We can use SNPs as an instrument to try and understand what's going on in terms of our disease outcomes. Okay, so I'm going to do a slightly odd thing here. Is I'm going to tell you the punchline to this joke first. When we look at whether or not early puberty, and this is in girls, affects hormone-sensitive cancers, we can see that actually it does. That there's quite a strong effect of earlier menarche timing on breast cancer, on endometrial cancer, ovarian cancer, and prostate cancer. Those of you who are paying attention will have noticed that very few people who go through menarche also then go on to develop prostate cancer. But this is because, as I showed earlier in the slide, the overwhelming majority of SNPs that are found for either voice breaking or for menarche are in common. So actually, when we talk about menarche SNPs, we're normally talking about puberty SNPs in general, though there are, of course, a few SNPs which are specific or different between the sexes. Now, we actually have a pretty good reason why this is happening. This is happening because it's about your lifetime exposure to hormones. And so if you have sex hormone exposure a year earlier, then you actually have an increased risk of cancer. It's simply about the cumulative exposure to sex hormones. And that's why there's this case for all of these reproductive cancers having an increased risk for earlier menarche here. Now, the problem with this slight situation is that actually there's quite a lot of overlap between the SNPs that are associated with puberty timing and the SNPs that are associated with BMI. So roughly a third of the SNPs that are associated with puberty timing are also associated with BMI. There's a clear metabolic link between the amount of adiposity that a person has and how early they go through puberty. From a sort of incredibly reductionist evolutionary point of view, puberty is effectively about when you start to reproduce. So your body has to have enough energy in it and in the environment to make sure that you can produce another human being. So monitoring of energy reserves and having energy reserves is a very important marker for puberty timing. and the move to the more obesogenic environment that we're seeing across certainly the whole of the UK, but more widely across Europe, is actually having a knock-on effect with puberty timing as well. So there's a public health issue, which is not just about this BMI thing, but also the effects of puberty timing. But because there's this overlap between the SNPs associated with puberty timing and the SNPs associated with BMI, that means that we might, as some of you may have come across in your MR studies, have an alternative pathway that links these SNPs associated with puberty to the disease. It might actually be that what we're seeing in our MR is driven by these BNI SNPs and not at all related to the puberty timing SNPs. So in order to try and counter this, we looked at multivariate MR. So this is where we take not just the association between those SNPs on puberty, but also where we consider the effect of those SNPs on, in this case, BMI. So we did a couple of different things here. Firstly, we did the sort of possible, sort of slightly naive thing where we just put the age menarche SNPs in a model. And actually, when we looked at breast cancer here, there was no association at all. And I think the first time we did this, we were slightly disappointed by this because we had hoped, we had this idea about the mechanism linking lifetime exposure to sexual hormones to breast cancer. We were reasonably confident that this was going to show something. But then actually we found a slightly unusual thing is that we split the SNPs into two scores. So I said about a third of them. So here we have 61 BMI related SNPs, which are also BMI associated, and 300 odd, which are not associated with BMI at all. And we split them into two different scores. And when we looked just at the BMI SNPs, we actually saw this slightly strong, slightly weird association, which I don't think even those people who work heavily on breast cancer have explored, certainly not something I've seen, where actually increased BMI appears to be protective for breast cancer, which is this slightly counterintuitive. It goes against all of the observational studies, but there have been certainly two other studies that have replicated this, that genetically increased BMI appears to reduce your risk of breast cancer. That's absolutely not true of the observational stuff. Increased BMI is definitely a risk factor for breast cancer, but there's something odd going on in terms of the genetics. But then if we consider those SNPs which are unrelated to BMI, so completely independent of BMI, we actually see what we expected, that we've got this lowering effect here where the SNPs are that raise your age of puberty, lower your risk of breast cancer. So the later you go through puberty, the lower your risk of breast cancer. When we sort of modeled these simultaneously, we got a result that was kind of similar to what we were seeing in terms of the just AMA raising alleles. So we're pretty confident that actually there is this true effect of puberty timing on breast cancer. If we consider a different cancer, so if we look at endometrial and ovarian cancer, these two together show a slightly different pattern. Here, there's not this slightly counterintuitive BMI effect. So actually all of the estimates, well, they're certainly sort of within the confidence intervals of each other, have a much more consistent estimate. So in both of these cases, BMI is probably a risk factor for these two cancers. So you get a much more consistent set of effects. Prostate cancer, however, looks like it's something that actually follows the breast cancer model slightly more clearly. Again, remembering that these are puberty timing variants. So we're not just simply looking at age of menarche, but we're looking at puberty timing in general. So we've got this thing that we're less pronounced than the breast cancer, but the BMI may in fact, genetically predicted BMI may in fact be protective for prostate cancer, but that boys who go through puberty earlier are at an increased risk of prostate cancer. So yeah, so prostate cancer may be something that looks slightly closer to breast cancer in terms of this slightly odd relationship with BMI. Now at this point, like I'm talking about public health, there might be a question about whether or not puberty timing is actually a target for public health, because most people, in fact, many people I go and talk to about this, feel that puberty timing is one of those things that's a little bit more fixed. I mean, they certainly understand that there's a genetic effect to it and then potentially environmental effects to it, but the idea of having an intervention that specifically targets puberty timing is not normally something that people immediately think about. So this is a graph, I talked about sort of like puberty timing in East Asia. So this is a graph from South Korea showing the change in puberty timing over about 60 years. So it starts in the 1930s and goes on to 1994. And there's been a very noticeable and very pronounced decline in puberty timing. Now this equally was happening in European countries, possibly earlier and possibly over a slightly longer time scale. But this change from people going through puberty on average between 16 and 15 and now somewhere between 12 and 13 is common throughout a lot of industrialized countries. If you think about puberty timing today, the average age of puberty timing is about 12 for girls and slightly older for boys, measured by menarche or voice breaking. And the reason that this is happening because there is a change in the obesogenic environment. I said that there was this tie between BMI and age of menarche, and the thing that's driving this is actually the food availability and the few nutrition in these primarily. So there's a major change in this. So this leads on, I hope, to a conclusion that actually many of the lifestyle interventions that we are already targeting at BMI are going to have a knock-on effect on puberty timing. And that if we target them at the right moment, we may get this double whammy of benefit. We may get a benefit because we've got increased metabolic health and the whole downstream list of diseases that affects. But also there's this reproductive hormone health that's happening at the same time. And that has benefits on reproductive cancers beyond what is normally associated with BMI. So yes, puberty timing, I hope, is a valid target for an intervention on a public health campaign. But if you think about what that intervention would look like, it's about many of the sort of campaigns that already exist to try and reduce childhood obesity, but targeted at an age when that will also have an effect on their puberty timing. So that was just a bit of a plug from me about the validity of puberty timing as an intervention thing, because many people sort of slightly overlook it. So one thing I was going to bring up slightly, so this is another one of the phenotypes that we look at, it's the age at which someone has their first child. I called it age at first birth, or AFB. And this is potentially an extension of what I was talking about in terms of those multivariate models, whereas before I was trying to tease out a specific effect of in that case, puberty versus BMI. Here what I'm looking at is I'm looking at something where I'm trying to understand a pathway. And the pathway I'm trying to understand is the effect of education on two metabolic diseases here. So the first is type 2 diabetes and the second is coronary artery disease, but actually they show a very similar pattern. So there's a lot of epidemiological evidence which is very strong that education has an effect on disease. And I think that has been shown both in so I've just noticed the chat. So I appreciate that I've flagged that I should be telling you to look in the chat, and I've only just seen that that's where it is. So let me just go through very quickly to see if there's anything. My voice isn't stable. I will try and get closer to this. Some of children associated with risk-taking. Yes, it is. The total heritability of early puberty. Unfortunately, I think the only estimates that are in my head are estimates from twin studies. So that's unfortunately probably about 20 to 60%, but almost all twin studies give an estimate of somewhere between 20 and 60%. I will see if I can get a more accurate estimate for puberty heritability and send you it as an email. And yes, you will get a set of those slides. The other factors involved in early puberty are, as I mentioned, the obesogenic stuff. So that's the overwhelming influence on early puberty. There's a few sort of monogenic disorders, but I don't particularly discover them, and those are certain things that are much more clinically relevant. And genetic variants increase reproductive hormones release cancer early? Yeah, absolutely. How big is the effect of MKRN3 on GnRH? I don't know off the top of my head. That's something, I'm not an experimentalist, so it might be that that's on the slide, but I don't know, unfortunately, sorry. So, this slide, looking at the association between education and type 2 diabetes and coronary arteries. Education, again, is something which there might be potentially an intervention. We've raised the school leaving age before. There might be a public health case for raising it again. But we were interested because when we started looking at age at first birth, there was quite a lot of overlap between the SNPs that we identified for education and the SNPs that we identified for age at first birth. So there is a link between reproductive timing in terms of when you start having children and the length of time in education. And that's seen in a genetic correlation very strong. I think it's about 0.7 between those two. And the top two SNPs for age at first birth are both well-known education affecting SNPs as well. So we looked at this in a sort of multivariate model. And actually we found that when we took the SNPs that have been known to be associated with education, there's about 600 of those, and we used this in a multivariate model, weighting by their effective age at first birth, that actually there was considerable attenuation of the education effect. And that was even stronger when we added in two additional phenotypes. So we added in age at first sex and age at smoking. Now, the reason we added these two in is because these are both categorized as risk-taking behaviors, but that they are happening specifically at the age at which people are coming towards the end of their education. So this is a sort of effectively a snapshot of their likely risk-taking at a very particular age. It's not necessarily the same as if you ask people if they speed on a motorway as they did previously, or if they ask them if they're generally a risk taker, because that's normally asked to people who are in their sort of 60s. But these potentially are getting to a much more specific sort of teenage, early 20s risk-taking type phenotype. And we think that's got quite a strong association with education. And you can see how actually when we add all of these in, certainly for type 2 diabetes, the two strongest predictors, and bearing in mind that this is using SNPs identified for education. So this is effectively an MR for education, but weighted for AFB and total smoking. But those are the two phenotypes that come across most strongly. So there's been a considerable amount of literature saying that education is a risk factor for all sorts of diseases. And yeah, it unambiguously is. It's got very strong univariate associations. But actually, when you start plugging in other phenotypes that might represent other behavioral patterns, what we see is potentially a lot of these associations are actually about a broader sociological trajectory, that it's not simply that sitting in a classroom for an additional year has any particular effect on your risk of diseases, but that there's a broad range of health-related behaviors together that might be having quite a big impact on what it is that's going on here. So we can use these sort of multivariate MR methods, not just to explain a specific risk factor for a disease and potentially identify something that we think is a good target for an intervention, but we can also try and potentially use this to understand a little bit more of what the causal web looks like here. Yeah, so that's potentially something that I think is going to be very useful in the future for trying to work out. The sociogenomic people are very excited about this as an option for trying to understand some trends in these sorts of data. So this is a final sort of thing when I'm talking about health. So ultimately, when we're talking about health, sort of one of the major arbiters of health is actually mortality. You know, the rate at which people die and the age at which people die. So one of the things that we can potentially do, and this is something that's become possible when we have studies on the same sort of size and scale of UK Biobank, and this is data that's in UK Biobank, is we can look to see what the effects of these variables are on ages of death. So this was a study which was effectively, for the first part, looking across single SNP associations to see whether there was a depletion of people at older age groups in UK Biobank, or for both fathers and mothers, and considering UK Biobank's reported parental ages of death. So UK Biobank asked people about what their father's age of death and their mother's age of death were. And actually, you can see that puberty timing variables are enriched in longer lived individuals. So they're sort of like, hopefully, ultimate arbiter of health, age of mortality. People who go through puberty later live longer. So rather than picking out particular types of diseases, we can actually see that in general, going through puberty timing is probably beneficial for a whole range of health. And we've actually seen in a sort of non-genetic way that if we look at the phenotypic associations, there's associations with cancers, with metabolic diseases, with psychological diseases, and just with how healthy people report on a sort of a snapshot type measure. So bringing all the effects of diseases together, there is this general feeling that actually puberty timing probably makes you more healthy over time. And puberty timing was actually certainly one of the top 3 strongest associations here, stronger than the effect on lipids or on heart disease. So I think puberty timing is probably very important across a whole range of different spheres of types of diseases. Ooh, right. So for the final sort of section, I'm going to talk a little bit about, you know, what can we learn from these sorts of GWAS studies about human evolution? And this is a little bit, frankly, more hand-wavy compared to the things I've been showing before. It's a little bit more speculative. But given that we're looking at phenotypes here, age at which someone had their first child, the number of children they have, menopause, the end of people's reproductive window, and puberty time, the start of that window, it's not necessarily surprising that we might be able to tease out a few bits and pieces here about what is going on in terms of human history and the sorts of selection patterns that have been observed across these sorts of phenotypes. So the first slide here is again, to start off on a genome-wide level. So we're not talking about any particular SNPs here. We're just looking at what the genome-wide level can tell us. And this is a study from a group in Queensland, which we're trying to measure this thing called S. And S is a Bayesian variable, which is effectively trying to model the idea that if there's strong negative selection, those alleles with large effects will have increasingly lower frequency. So it's modeling a combination of the effect sizes of each of the SNPs combined with the allele frequency of those SNPs, and just trying to understand what the shape of that association looks like. And those phenotypes which have a large number of SNPs with very small allele frequency that are the largest effect sizes are likely to be those that have been under negative selection. So if we look across all the various different phenotypes, they token, I've highlighted 3 of ours of particular interest. So they have age at first live birth here, potentially actually slightly weaker than I might have thought, but definitely strong evidence of negative selection. Age at menarche, again, strong evidence of negative selection. And finally, age at menopause was actually their strongest phenotype. It's the phenotype with the best evidence of natural selection. And again, this idea that menopause is the end of your reproductive window. You know, beyond that point, you cannot have more children. The fact that there may be a negative selection here is perhaps unsurprising. But this is a kind of blunt tool. It tells you about phenotype. It doesn't tell you anything about any of the particular SNPs. So I've got a couple of slides left, which may tell you a little bit more about what might be going on at particular sites. So the first of them is that we were pleasantly surprised to discover that we had a number of variants when we were looking at puberty timing, which were in imprinted regions. Imprinted regions are those where there is methylation that occurs that affects the transcription of various genes at those sites, and that methylation is applied in a parentally specific way. So sometimes it's occurred if that particular site of the chromosome has been inherited from the mother, versus applied whether or not that particular site of the chromosome has been inherited from the father. And we had a couple of regions here. You'll remember MKRN3 from previously in the talk, the break on GnRH secretion. We also had 2 in GLK1 and 1 in MEG9. And these black dots here are roughly what our normal GWAS looks like. So when we ran a GWAS, this is the, but we were very lucky to be able to get data from 40,000 women from the DECODE study where they actually knew what the segregation of those chromosomes looked like. So they knew whether they had come from the mother or the father. And when they ran it in their study, we got this. So in each of these cases, one of the alleles has an effect when it's inherited only from a very particular parent. Now, unfortunately, it's not the case that we only see an effect when they're associated by the maternal, or only see an effect when they're associated by the maternal. We see this as balanced. So for 4 out of the 5, it's the parental inheritance that has an effect, and for 1 of them, it's the maternal inheritance that has an effect. Now, it's a bit difficult to work out exactly what's going on here with the effect allele, because we don't necessarily always know whether it's that allele that's associated, we just know that it's that site that's associated. So this allele inherited from the mother in MEG9 could have had an effect either positive or negative, depending on which allele it was that was associated. But it's certainly true that puberty timing is something where we do see these imprinted parent of origin specific effects that are occurring in important regions, some of which where we have good biological knowledge, such as MKRN3, across the genome. Now, why am I talking about this in terms of a sort of presentation about evolution? Well, there's quite a good evolutionary theory to suggest that the reason that imprinting has existed in mammals is almost this sort of like battle of the sexes idea, that there may be situations in which for the mother or the father, from a fitness point of view, there are different optimal strategies that are going on here. And often those are focused on early life exposures, because again, the theory about this is in an incredibly reductionist way, that because the mother puts more resources into the child, it might be that that mother wants each of the children to get the resources fairly, whereas the father...
It's windy out there, huh?
Sorry, is that me being inaudible? But when it comes from the father, they may want the mother to give as many resources as possible to their offspring. And if, again, very reductionist, you think of puberty time as effectively the culmination of childhood growth. This is the point where you have absorbed all the energy that you need to be an adult, to be part of your reproductive window. Then actually you can conceptualize timing of puberty as a good measure of the amount of resources that you might have got from your parents up until that point. So to have this as effectively a model of what we think this battle of the sexes in terms of allocation to children might be, might make some sense. And so far, it is, I think, true that puberty timing shows the best evidence for imprinting effects. There are certainly some for birth weight, less obvious than they are for puberty timing, but for other adult-related diseases, for example, they're nowhere near as strong. Puberty timing is definitely something where there's a good suggestion that there is strong evidence for an imprinting effect. So it suggests that actually puberty timing from an evolutionary point of view is something where there has been this differing in the optimal strategy between parents, and that potentially tells us something about human history. So that was a little bit about both a genome-wide level and then a slightly more focused level about a particular set of SNPs. And I'm just going to, and this is my final set of examples, talk to you a little bit about FADS1. So FADS1 is something that we identified in a GWAS study for a number of children. It was not the only thing we identified. We identified quite a lot of variants for this, but we were quite interested to identify FADS1 because FADS1 has got a long history in the evolutionary biology literature of being an important site of selection. It's one of the strongest sites of selection in the human genome. Now, FADS1, in terms of its sort of like biological processes, has been quite well studied, and it's involved in the breakdown of long-chain polyunsaturated fatty acids. And the theory behind FADS1 is that there was an important change in the selection between the movement from a meat-based diet of hunter-gatherers to a more vegetable-based diet of farming in human history, and that actually there was an advantage to being able to digest fatty acids from different sources, and that gave people an advantage. Except we find it actually today in a study that was done on people in the 20th century for their number of children. Now that suggests actually that there's something going on here in terms of a particular specific relationship to reproduction, not necessarily something about your digestion of fatty acids, because the majority of people in UK Biobank in which this study was done are not, you know, hunter-gatherers versus farmers. There's not that sort of disparity in this data. Now, what might be going on here? Well, this is sort of like an overview of what I'm talking about in terms of the change in selection. So this particular allele, and it is the same allele in both of these plots, has over the 1,000 years in the historical allele frequencies changed quite markedly from about 10% to about 65%. And it varies within human populations today, but there has been a very clear and obvious selection over the data forecast. Now, this I have to stress before anyone gets too excited, is non-significant. But it is certainly and undeniably a sort of a direction of travel between people born in the 1940s compared to those born in the 1960s. And that's just within UK Biobank. So the frequency of this allele could well be increasing. And the fact that we find this for the number of children you have suggests that actually this is something that is actually going to be increasing in subsequent generations. Now, in order to sort of try and understand this a little bit more, we looked a little bit further into this allele, and the classic thing is, well, if previous people thought it was about fatty acids, what happens if you look at genetics that are associated with those particular fatty acids? And actually, FADS is the only one that shows this particular pattern. The other classic long-chain fatty acids variants show no association with number of children born. I mean, is it something that's about an HDL or LDL effect? Well, again, none of the other SNPs associated with HDL or LDL show this sort of pattern. So we're not really sure what the mechanism is going on here. It's something we're going to look into further. But I think what I will hopefully leave you with is perhaps the slightly confusing interpretation that there might be something which is affecting reproduction both thousands of years ago and is still potentially visible today. And that it's not that this change was entirely driven. It may have been primarily driven, but it wasn't entirely driven by a change to people's diet, that there is something specific to reproduction that is also happening here at the same time. So, a bit of a summary. So, the first thing to say is that reproductive traits are highly polygenic and with complex architecture. And I'm hoping that over this course of this week, you've seen this is something that is true for any complex disease or phenotype that you might wish to understand, and that by having this sort of highly polygenic, lots of SNPs, we can actually start using some interesting downstream methods. We can start to explain enough heritability to be able to do Mendelian randomization, or also those sorts of genetic correlation type things that I talked about earlier. to try and understand the interplay between two different diseases or a risk factor in a disease and try and understand the shared biology that's going on between those. That we might be able to actually start to understand some of the biology that's underlying these conditions. So even if we don't necessarily identify a risk factor, we might be able to identify a drug target or a pathway to particularly implement. And that can be something that's really quite clear and specific in terms of the mass of SNPs that are showing us, similar to the DNA repair from menopause. Or it might be something that's only affecting a particular set of SNPs, such as the mkRNA break on a GnRH secretion. And we can hopefully transition some of the things that we find in GWASs into these animal models to try and understand them a bit better. And then finally, and again, this is definitely a bit more hand-wavy, that we might be able to get some insights into these phenotypes, into what has happened in human history and the sort of pressures that might have affected our ancestors in terms of how we got to where we are now. So with that summary, these are far too many people to actually acknowledge, but these are all the people who had helped me on many different studies that went into this. I feel like I need to at least show their names so that they're sort of acknowledged slightly. But there is definitely a core of people that I particularly like to thank. These include colleagues at the MRC epidemiology units throughout Cambridge more widely, in Erasmus, in Boston, and at Decode and 23andMe, and over in Oxford, who have helped with many of the sort of more sociological things. So thank you very much for listening, and any questions?
Thanks very much indeed, Felix. I guess you already dealt with most of the questions in the chat, but if there's any extra ones in the chat, or if people want to